What if the future of medicine lies not in discovering new drugs, but actually engineering them with precision and safety at the forefront? Well, today I'm excited to have Chris Hart, the CEO of Creyon Bio, join me on the podcast, a company that is leveraging AI to revolutionise drug development. And this approach has led to remarkable breakthroughs, including rapid development of a treatment for a baby boy with an ultra-rare gene mutation, an achievement that took 13 months from concept to dosing. I want to know more about the real difference that technology made for that little boy. And my guest is also going to bring a unique perspective to the intersection of AI, biotechnology, and personalised medicine. And his experience in using AI to decode complex data and optimise chemical modification for OBMs is not only innovative, but also represents a significant shift in how we think about drug development. So we'll talk about all that, how it could potentially shorten drug development timelines, increase success rates, and change the way that we treat rare and complex diseases. So, what are the implications of AI in drug design? How could this technology reshape the future of medicine? These are a few things we'll explore together today. Delivering daily content to 140,000 of you wonderful monthly listeners across the globe is no small feat. I don't want to take all the credit here because it wouldn't be possible without the backing of our dedicated sponsors and partners. And I also want to shine a light on the fact that legacy managed file transfer tools are looking dated. They often lack the security that today's remote workforce demands. And companies that continue relying on that outdated tech, they're in danger of putting their sensitive data at risk. Attention IT professionals, are you tired of juggling these multiple servers for secure file sharing? Integrated shared folders and email, plus a comprehensive REST API. Kiteworks simplifies your workflow. For administrators, you can experience unmatched functionality and integration that traditional MFT servers just can't touch. Step into the future of secure managed file transfer with Kiteworks by going to kiteworks.com to get started. That's kiteworks.com. And remember, Kiteworks is also FedRAMP moderate authorized. Thank you for your patience today. This is the moment you've been waiting for. It's time to welcome my guest onto the show. So enough from me. Let's get today's guest on. So a massive warm welcome to the show, Chris. Can you tell everyone listening a little about who you are and what you do?
Great. Thanks, Neil. Very excited to be on the show. Thanks for the opportunity. I'm Chris Hart, co-founder and CEO of Creyon Bio. Just a bit about me. I'm a scientist by training. My academic training is really at the interface of biology and computer science. And after I finished my doctorate work, I moved on and I bounced around between a variety of posts in biotech, pharma, and even spent some time doing science policy.
Well, it's a pleasure to have you on the podcast with me today. What put you on my radar was when I first discovered that Creyon Bio uses AI to engineer Oligonucleotide-Based Medicines, or OBMs. And just there's so much hype around AI and things now. But how are you seeing AI enhancing the drug development process, particularly in ensuring safety right from the outset? Because as I said, we hear a lot about the hype, but this is real problems we're solving here, man.
Indeed. And just to take a step back, because I think it'll help contextualize how we're using AI and machine learning in drug development at Creyon, it's probably good just to give the listeners a little bit of a background as to what oligonucleotide-based medicines are. Sometimes I'll refer to them as OBMs because oligonucleotide-based medicines is a bit of a sort of a long phrase. But OBMs are, the way to think about them is they're effectively DNA-like molecules, short DNA-like molecules that we chemically modify to improve their drug-like properties. And one of the things that makes them remarkable as a class of drugs is that they are foundationally different than other types of molecules. The first big difference is that they're polymeric, so they're different than small molecules. But even amongst polymeric drugs like peptides or proteins, OBMs are special in a different sense, which is both peptides or proteins or antibody drugs, they all interact with things in the cell to have pharmacological effect through the complex interactions of really how the drug interacts with these biomolecules. And OBMs foundationally work by binding to the information molecules in the cell, either RNA or DNA, and in our case, largely RNA, through Watson-Crick-Franklin hybridization, which is A's to T's and G's to C's that we've all learned in probably high school biology, which makes them effectively, quote unquote, information drugs. So we're no longer trying to sort of make and connect really complicated puzzle pieces where we have to understand the structure of a protein and how a molecule is going to interact with that complex structure and the complexity of a cell environment, but rather we can leverage the information system that the cell uses all the time. And you put that in the context of where we live today in a sort of an arc of technology. About 20 years ago, we sequenced the first human genome, and it was a race between a private sector and the public sector, and they declared victory, which was pretty amazing. And what that victory brought us from a science perspective across really medicine at large is we now can unravel the genetic and molecular basis of disease better than ever before. And we know often what genes need to be controlled to have therapeutic benefit. And oligos allow you to have a direct path to making that control. The other piece of the world of oligonucleotide-based medicines is it's actually a pretty mature field. It's been sort of in the shadows of drug development for a while, but the first sort of seminal experiments that sort of showed this was a feasible idea came into the light about 40 years ago. And over the last 40 years, what's really emerged is that you can make good drugs out of these chemically modified DNA-like molecules. They can be blockbuster drugs. But what has been slowing the adoption of this medicine is that the hope that it would be a really fast and easy translation from information about an RNA molecule that you want to control to a drug because it's just right to its first complements. It turns out that's easy. But when we put these chemical modifications onto the types of medicines we make, these molecules often end up being toxic or inactive. And that complexity is built into the frame of how do you assemble these polymers. And what we recognized at Creyon is these are polymeric drugs, which means they're like other polymers. We should be able to engineer these compounds like we engineer every other polymer that we depend on every day. But the problem is no one had created the right data to really understand the complexity of how the different components would interact. And when you do start to tease that information out by creating the right data, you also recognize that this is a very complicated landscape with many intricacies and complexity. And this is where AI and machine learning comes to bear and has just huge potential to help really understand these complex data spaces. And that's what we've been leveraging AI for, is really how do we first create the right data, and then we can feed into these models, and these models can help us understand the data so that we can walk out not with simple heuristics, but rather with engineering insights so that we can create these medicines much more facile than we could if we were trying to use traditional trial and error-based methods.
And as someone that has lost a child to a rare disease, your recent success in treating a baby boy with an ultra-rare disease is nothing short of remarkable. So can you tell me a little bit more about this case and also the role that AI played in developing the treatment?
Of course. Yeah, this was a pretty amazing story. And it really is a hallmark of where we're headed in terms of modern biology and what it has to sort of offer to this notion of translating the genetic understanding of disease to novel therapies. And in this case, this is work we did in collaboration with the TNPO2 Foundation. And the TNPO2 Foundation was established by a family whose son was born, unfortunately, with a variant in the gene TNPO2, transportin 2. Just to give you the context, transportin 2 is a gene that encodes a protein that's important for cellular function, in particular neurons, and it works to keep things in and out of the nucleus, which keeps neurons working well. But the story of our collaboration really started when Leo was born and the family and the clinicians realized he wasn't thriving, and they had an opportunity to get him sequenced. And they sequenced his whole genome, and they diagnosed him with a very rare disease. In fact, the disease had only been identified in one other individual in the entire world ever before. And it was, in fact, a variant that was a single nucleotide polymorphism, which is to say a single A, C, T, or G was changed from what it is in all of us to a different base in this gene, a 1 in 3 billion error, if you will. And because we live in the world of modern biology, we're able to look at that data and understand that the variant there was likely causing that gene to be misfunctional or dysfunctional. And it was likely causing a production of a protein that wasn't just broken, but rather causing additional toxicity. It'd be just an analogy I sometimes like to use is if you made tires at a factory, and in this case, the tires are proteins coming off of a gene, and your factory made 50% of the tires to be broken, right? They just couldn't hold air. And then you shipped all your tires off to the car manufacturer. Pretty much every single car would come out of the lot broken because it's really unlikely if 50% of your tires are broken that you'd end up with 4 tires that were working. And this is effectively what was happening inside of Leo's cells, is that where they were making 50% of this TMPO2 protein was bad, and it was causing aggregations of effectively this misfunction to build up. But the sort of tire analogy it holds is also the sort of analogy to tell you how you can fix this, which is if you just got rid of, before you sent those tires out to the car manufacturer, the bad tires, if we just got rid of the bad TMPO2 gene, the good gene, as long as you had enough of it, would suffice. In the car analogy, maybe you'd make slightly fewer cars, but you'd still have functional cars. And in this case, also a testament to modern biology, we knew that there are people in the world, because we've sequenced millions of people now, that actually live just fine with only one functional copy of TMPO2. So that gave us confidence that there'd be something to do. So, and it gave the family confidence as well. So they were diagnosed when Leo was just 3 months old. And then about 7 months later, they found us. And they found us having this information in their backpack, if you will. And when they found us, we were also in a fortunate place to be able to help. We had just finished putting together the final touches on our first iteration of our platform. And when I say our platform, this is a sort of a collection of data, machine learning tools, and assays that allow us to very rapidly go from a gene you want to modulate to a drug that would be expected to be safe and active in people. And the tools we need to validate that assertion would be right. So when they came to us, we said, yes, we'd help. And ultimately, what we ended up doing is we used the first iteration of this platform, which worked as we had all hoped it would, and it allowed us to go from really whiteboard to a lead drug that we were ready to scale up for carrying all the information you need to sort of get approval from the US FDA to dose the patient. And that first worked from engineering to validating work in both patient fibroblasts and animal models to get to a place where we were ready to scale, to get to a drug product, if you will. All took us 5 months. And for those who aren't in the sort of drug development world, that's remarkably fast. And on top of that, we did it both at breakneck speeds, but also we tested in total 96, but of the first 48 we tested, we ended up with 8 leads that came out that were all like great drugs. So we had to choose one. We scaled it up for what we call good manufacturing processes, which you need to make drug products. And then we did the GLP studies, which are good laboratory practices. And then we filed for an application with a clinician that was going to treat Leo, and an investigator-initiated IND. And the FDA gave us the go-ahead to treat Leo, and we started treating him last July. So he's been on drug now for over a year, and I'm super excited to report that in this process of engineering a drug, validating, and then getting approval to dose a patient, all at breakneck speeds of less than 13 months to go from whiteboard to dosing, we also ended up making a drug that's having significant benefit for Leo. He had, even in the time frame in which we met the family, his development of milestones started to slip, and his severity of seizures started to get worse. So by the time we dosed him, he was having seizures, often seizures that would stop him from breathing 6 to 12 times a week, if not more frequently. And he had lost his ability to really engage with the world. He just couldn't control himself. He couldn't focus his eyes, and really was sort of non-responsive to the environment he was in. And then we started dosing him. And then we started at a subtherapeutic dose to make sure that we're being cautious in case there were some adversity that we did not pick up in our preclinical work. Fortunately, that did not happen. And by the time we got to the fall, we were able to get to a therapeutic dose, and we started to see real benefit. There was a precipitous drop in his seizure frequencies. We saw milestones regain, and he's clearly better off than he was not on drug. And that is continuing to be an improvement that we have seen throughout the year. And the FDA actually just gave us the sort of go ahead to increase the dosing frequency, even because the drug has been so well received. The high notes there is that on top of making a drug that's having tremendous benefit for this patient, we also demonstrated the platform allows us to make safe and effective drugs hyper efficiently. And it allowed us to make really the first locked nucleic acid antisense oligo that's an allele selective drug that's ever been administered to a patient. And that's actually a pretty big deal because LNAs, these are chemical modifications that were discovered in the 2000s-ish, and people hadn't been able to apply them, even though they improved the activity of drugs significant tenfold. People have been able to apply them in the clinic as readily as they would have liked to. In fact, there's a splash of publications that came out in the 2010s era associated with this type of chemistry where people said they were just flat out toxic, which was 100% wrong. These drugs are not toxic, carte blanche. They're only toxic if they're put on the wrong sequences and in the wrong configuration. What the platform we developed is it allows us to actually leverage these sorts of modern chemistries in a way where we can control the safety from an engineering principle point of view and not rely on trial and error screening, which allows you, where you fail, to actually be able to identify these safe compounds. So this drug is actually a first of many kinds, and it's also the first of many that we'll be able to create off of the platform we developed.
Absolutely phenomenal what you've achieved here, and a big shout out to Leo and best wishes from me to him and his family. And I'm curious, how does Creyon Bio's approach to drug development, how does it differ from, let's say, more traditional methods? And what significant advantages does AI bring to identifying safe and effective medicines?
That's great. So I think the way you can think about it is that we're truly engineering rather than discovering molecules. Traditional drug development has always been predicated on this idea, which is it's hard to find bioactive molecules. And that sort of makes sense. Your cells are robust. Finding a molecule that interact very precisely with some biological process to have therapeutic benefit, that's challenging. We've gotten better over the years with high throughput screening and now generative AI methods to sort of identify these complex interactions that may be helpful in driving these things. But at the end of the day, that entire workflow is built on this notion that the serendipitous moment for drug discovery is when you can find a molecule that interacts with another molecule to have benefit. And that interaction is the thing you drive for. And in other words of saying that, they look for activity first. And then once you show something's active, you'll go forward and you'll say, great, I hope this thing is safe. And often it's not. And then often you'll spend the next iterations, which actually turns out to be sometimes years to decades, and sometimes it'll cost hundreds of millions of dollars to iterate on that process, hoping you can find something that would be well tolerated enough to go into the clinic and retain the same activity. What we've built is something that allows us to use oligos, that allow us to sort of avoid and skirt the problem of having to figure out how we're going to interact with complex three-dimensional structures. We just interact in the way that information molecules in the cells exist through hybridization. So that's the first thing. But the second thing is, how do we make sure? So activity was never really a major problem, or binding was never really a big problem in the space, but the sort of assurance of safety was very much the challenge in the space. And what we've built out is a way of actually understanding from data and then the machine learning models that allow us to then deploy them with sort of engineering crispness, where and how we should put chemical modifications in these molecules based on the sequence of them, such that we can have a very high expectation that they would be well tolerated. That has huge implications. Right now, we've cut out this chunk of early drug development, which is both failure prone, costly, and slow and unpredictable largely. So when we think about the process, one of the things that we're systematically trying to do at Creyon is erode the cost and time it takes to create new medicines. And this was the first thing we had to solve to make sure we could expedite the process of going from target ID to a lead that we could push into. And AI was a critical piece of this, because the complexity of the design world associated with oligos is it's immense, and the interactions of the components that make up oligos is also highly complicated. So this would be an untenable task if you were sort of trying to write down simple heuristics that you could understand and sort of just draw on, like a drug should only have, 3 of these in a row or 2 of that. That's inadequate. The way you have to think about these is in a much more holistic way, which allows you to sort of take into the complex correlations that exist between every aspect of the molecule.
And I think the intersection of AI and medicine is rapidly evolving field. And I'm curious, from what you're seeing, what are the particular trends or maybe even advancements in this area that excite you the most? And how do you see them shaping the future of biotech? I understand it is very early, but what particularly excites you at the moment?
Yeah, it's a great question. I think AI is, I'm excited about many aspects of what it will bring. But in biotech, one of the challenges that you face is that data is often sparse. So in the near term, a lot of the things that you think about where we're going to see advances are going to be in places where we have access to data and where there's opportunities to leverage that data in novel ways to gain insights that'd be difficult for us to do as mere mortal brains because we can't hold that corpus of information in our heads as well. So when I think about that, I think about what and where I'm really excited because it's going to have huge impacts are in places like understanding disease at a resolution where we could actually have preventative bell towers, if you will, to let us know that we're walking down places without having expensive interventions with biomarkers. And a lot of people think about biomarkers from some what sort of things you can measure out of the blood that would be a signal of disease. I think we see, I'm wearing a smartwatch. Neil, I can't see you, but you may be wearing a smartwatch. The amount of information that we can now get off of these digital tools that allow us to really understand at both a temporal basis, at an individualized basis, and at a cadence that allows us to see perturbations from normal and then connect to what would be expected for you personally, because we're all somewhat different based on the corpus of data that people can collect. I'm very excited that we'll be able to sort of have these biomarkers that come out, especially digital biomarkers that allow us both have earlier diagnosis that things are going amiss. So we maybe can have earlier interventions. But in addition, that clarity also allows us, when we're thinking about developing novel therapies, to understand in more clarity sort of when an intervention is having benefit for patients in a way that will allow us to have, hopefully, perhaps trials that, especially in the rare communities, don't have to rely so deeply on placebo controls, or we can get away with shorter timescales, or we can get away with even just higher resolution of which patients under what conditions would have benefit for the drugs we're pushing forward. Along those lines, that falls into allowing us to do things like build out digital twins or having better diagnoses, and sort of, I think that'll also fall forward into understanding really the systems medicine arena around disease pathologies.
Completely agree with you. And I'm currently testing a smart ring, very similar to the smartwatch there. And it really got me thinking about how we're moving towards more proactive approach to healthcare rather than the reactive approach of waiting for something to go wrong. And for you guys, I mean, with the ability to engineer drugs for safety first, how does AI influence the speed and efficiency of bringing these new medicines to market? I know we've talked about this a little already, but is there anything else you can share around that on influencing that speed and efficiency?
Yeah, we often think about it just in terms of, this is not new in the field, obviously, but thinking that one of the challenges in drug development generally is that from idea to drug in the market, timeframes is both one way of thinking about it is that's the biggest problem, which is it costs too much to make too few drugs. What's the driver of that? It's really, we have a lousy probability of success from project initiation to making a drug. We think a lot of the work that we're doing is really sort of foundationally pushing it forward on the way of how do we improve the probability of success in that process. And the way that we sort of started where we are, and the reason that we started where we were, was that when we thought about it, what would be the biggest impact on improving probability of success and hopefully sort of mitigating costs in terms of time and real costs to make drugs so that we can get rid of this problem. If you look across the industry and ask, why do drugs fail? There's really, in broad strokes, there's a handful of reasons, not many. One is they fail because they have safety liabilities. They fail because the therapeutic hypothesis is wrong. You're modulating the wrong thing. It just doesn't help. You made the wrong drug. It doesn't do what you thought it was going to do. And the last one, which people don't always think about, is that your drug doesn't actually get to where it needs to go to have a therapeutic benefit. So when we think about what we've built, is we've built a platform that really foundationally resolves the safety liabilities. How do we connect data together from the corpus of information we create so that we truly have, for every drug we make, an improved probability of success in terms of safety? I think we're on track doing that. We're creating new data every day, and our platform allows us to integrate that data in ways that others just can't. And it's truly a platform. It's not a set of learned heuristics, and it's not hope that our magic chemistry is going to make our drugs better, but rather it's data-driven systems that allow us to truly understand which chemistries under which contexts are going to have benefit to improving safety. The second thing I mentioned was we were going after the wrong therapeutic hypothesis. It's been long established in the field that if you're targeting the genetic basis of disease, you actually have a better chance of success generally, because you're actually going after something that has sort of a causal error, if you will. So oligos give us that advantage already. And we know because oligos all work through generally the same molecular actions, we're unlikely to create the wrong drug. It'll certainly do what we want it to do at a molecular level. So those are the first three things that I think we've sort of been addressing with the platform and our choice of substrate, if you will. And the last one, delivery problems, I didn't talk about, but this is really the sort of the other Achilles heel of the industry is that oligos don't have broad tissue access by default. So what we've also built forward is a way for us to improve the distribution of oligos into tissues they'd otherwise not be able to have access to. And we do that by leaning on our core competency around engineering nucleic acids. And we use a set of technologies called aptamers. Aptamers are effectively chemically modified nucleic acids that rather than interacting with the sort of their targets through Watson-Crick-Franklin hybridization information code, if you will, they actually do form complex three-dimensional structures that can bind to receptors on cells. And what I think is interesting about that is that we can use them, but unlike trying to do that to actually control the molecule, we're only looking to bind to that molecule, and we're only looking to bind to that molecule long enough to actually bring our payload that can control the gene expression into a cell, which is actually a much simpler problem than if you're trying to do that sort of interaction to create a novel therapy. And what we've built is a set of tools that allow us to do that very efficiently. So now we've solved what I think are the foundational pillars that often drive drug failures, and by improving the probability of success, our hope is that we'll have an outstretched sort of capacity to improve the ways in which we can create novel medicines for both rare diseases, ultra rare diseases like we did for Leo, but even common diseases as we push forward, as the processes are the same, regardless of whether you're treating a patient has a bespoke variant that's driving their disease, or you're treating a disease that's fairly common in the population.
And I'm conscious we might be making it sound very easy, but I'm sure there's been a lot of challenges along the way. So what initial challenges did you face when integrating AI with traditional biotechnological practices? And how have you at Creyon Bio overcome some of these obstacles? Any challenges you can share there?
I think I alluded to this before. The biggest challenge, I think, it was twofold. One is data, and the second, everything we do is dependent upon having an exceptional team of people. And the work that we do is challenging because it actually spans a broad set of domain expertise. So finding people that are domain experts and then making sure that we are able to assemble teams that can work together well and span these disparate domains that need to be spanned is something that we've spent a fair amount of time creating. And I think the team's spectacular. And we're constantly working in frames where we have deeply quantitative scientists working with bench scientists and these dry scientists and wet scientists working together with across domains, whether it's chemistry, pharmacology, computer science, mathematics, physics. That teaming is critical. The second challenge that I think is obvious is that data is king here. So we spent the last almost 5 years now at Creyon. The vast majority of that time was thinking about how do we create the right data to build models? Because what we recognize is that retrospective data for many AI applications, like if you want to build a large language model to predict your next word or your next sentence as you're writing, or help with a variety of things to render pictures, that's great because you have terabytes and terabytes of data. If you want to engineer what should be your next drug, you don't have that luxury because there's just not that much interconnected data. So we had to think very deeply about how do you think about creating smart data, not just big data.
And I think the other area we've not discussed is how regulated the industry is. It's an incredibly highly regulated industry. So how do you ensure that the use of AI in drug development aligns with some of those regulatory requirements, particularly in the context of developing treatments for rare and ultra-rare diseases? Any obstacles here, or has the community been largely embracing of it?
I'm pretty excited about the tails of the sail, if you will, from what regulators are working on. It's an emerging space in terms of how does AI feed into the drug development, but both FDA and agencies at the UK and Europe are actively pursuing both seeking public comment and feedback from industry about how can we leverage AI optimally to help sharpen our regulatory processes. So I think we're on a path here, which is good. And it's going to be a true collaboration between regulators and clinicians and patients in the industry to sort of figure out what is the best way so that we can do what we need to do, which is assure safety and efficacy of these types of medicines we make, while also leveraging the tooling that's coming out from novel science optimally. So I think that's an evolving space that's moving in hopefully what will be a continually positive place. And then I think from the sort of regulatory space around rare versus ultra rare versus common diseases, that's also a place that I think that regulators have been very forward thinking in many spaces where they've made, I would say, simplifying processes to enable rare disease development where it would be otherwise untenable. The work we did with the TMPO2 Foundation treating Leo, we were able to leverage, for instance, the N-of-1 guidance that the FDA put forward to allow us to sort of have an expedited path through the agency through an investigator-initiated application. I think there's work to be done there to broaden that out, to sort of see how we can connect those advances to industry such that there's viable commercial models to allow for stronger innovation. But I think the sort of momentum in that space is going in the right direction. And we're always excited to work with policymakers and regulators as we do the work we do.
And looking ahead, what are Creyon Bio's goals for the next few years? How do you see AI continuing to transform the landscape of drug development, personalized medicine, and so much more? There's so many big opportunities there, but how do you see that taking shape? What are your big goals there?
Our next couple of years will hopefully be marked as successes of taking the platform we've developed and using it to both advance our internal programs, and we also have partnerships with other biotech and pharma companies that we're excited to work through to sort of advance compounds into the clinic, not just for N-of-1 diseases, but also for common diseases. As those go forward, we're very excited to have an increasing set of data that connect the work that we've been able to do to really engineer compounds for safety as far as we can tell with preclinical readouts. But as we get clinical readouts, those readouts that we've done preclinically will have clinical connectivity to will, and that will be very meaningful for us. We'll be able to sort of further enhance the platform for predicting out and having validation that the readouts that we're making, the predictions we're making in terms of thinking about preclinical safety correlate to additionally, and most importantly, clinical safety. And that'll be great. In terms of extensions on the platform, we're already, I think, about 100 times more efficient. We're going from target to lead than others in the space. I think we're on track to add another order of magnitude to that over the next couple of years. And if you think about what that means, really, it puts us in a place where our in silico approaches are actually starting to perform as well, if not better, than any known preclinical assessment of a drug before it goes to the clinic. And just to give you context, right, most drugs that go to the clinic, they start at phase 1 trials, which are mostly safety studies. About 50% of those drugs fail in safety studies. So whatever we're doing to assess drugs from a preclinical perspective, when I say we, just to be clear, I'm talking about the industry at large, Creyon, and that's better than, of course, not being 50-50, but we have some room to do. But if our models actually allow us to do as well as that, think about the implications of what that means as you think about translating genomic discoveries into new medicines. Is if you truly can engineer compounds where you have the same probability of success going into the clinic as you would after spending a decade doing preclinical research on sort of an activity, then trial and error safety sort of optimizations, and then going into the clinic. We think that ultimately will have a huge potential in terms of allowing us to really leverage the information that's come out of the post-genomic world in a way that's quite exciting, such that we can really sort of start treating the unmet medical need that exists that we know how to correct. So that's, and I think where we are with Creyon is we have a sort of a stepwise path to get there while also creating novel therapeutics that meet unmet needs that can sort of fit into the development processes that exist today. So we're systematically trying to walk through this process to really improve the efficiencies of drug discovery. And in the long run, I think it'll be pretty exciting times.
Exciting times indeed. And I can't thank you enough for taking the time to come on here and share your insights with me. But before I let you go, I'm going to ask you to leave one final gift to the listeners, and that is either a song that means something to you, we can add to our Spotify playlist. Guilty pleasures are allowed, or a book that we can add to an Amazon wish list. What would you like to leave and why?
Oh, can I leave more than one book, or do I have to leave one?
Go on, I'm feeling generous. Go on, what you got?
All right. So, the sort of, I think about, I try to read what I can, but the two categories that I think are really interesting is, one is contextualizing history. I found both, and a lot of folks have seen this, but Sapiens as being a really interesting book to contextualize sort of where we are today. But then another book, which doesn't have quite as much fanfare, is The Ascent of Money. Both of those books are really interesting lenses to look at sort of history through, which didn't necessarily reflect in my academic work in history as I was going through grade school and high school. And then from a company perspective, I found this book about Bell Labs, The Idea Factory, quite fascinating, about how the Bell Labs was able to sort of cultivate a community of really constant innovation. And they set the stage for where we are today with modern electronics and really the computer era and subsequently the sort of internet age. And then I really enjoyed the books around Amazon, both Everything Store and the sort of collection of shareholder level letters that Bezos put together and Invent and Wander. And then from a company creativity and culture perspective, I thought Creativity Inc. is pretty amazing book, actually, in the way that they do evaluations of early movies to hone the story with everyone's feedback.
Awesome. Well, I will get all those books added to the Amazon wish list. And for anyone listening, just want to find out more information about you, your team, or just dig a little bit deeper on anything we talked about today, anywhere in particular you'd like to point them?
LinkedIn is probably the best path. You can find me if you search for Christopher Hart. You'll find many Christopher Harts, but I'm the only Christopher Hart at Creyon. So if you search for Creyon and Christopher Hart, you'll probably find me. And then our website, creyonbio.com, is a good place to learn more about what we're doing.
Well, I've just loved chatting with you today about the role of AI, especially treating the baby boy with the ultra-rare disease there, and also sharing insights on that intersection of AI, medicine and drug development. They're both valuable as they are compelling, but just more than anything, thank you so much for taking the time to sit down and share your story today.
Well, thank you, Neil. It's been great to chat.
So as we conclude our discussion with Chris, I think it's clear that this company is at the forefront of a new era in medicine. And by integrating AI into the very fabric of drug development, they're not just making the process more efficient, they're changing how we approach the treatment of rare and complex diseases. And the story of baby Leo's treatment is, for me, an incredibly powerful example of what is possible when cutting-edge technology meets a deep commitment to patient safety and innovation. And as I reflect on the conversation today, I think the potential for AI to transform not just drug development timelines, but also the overall success and safety of new treatments seems to be something incredibly magical happening here. But how might these advancements impact the way that we approach healthcare, the way we approach disease treatment in years to come? These are a few of the things I'm going to be thinking about. Remember, email me, techblogwriter@outlook.com. Twitter, LinkedIn, Instagram, just @Neil C. Hughes. Let me know what you're going to be thinking about after this conversation today. I hope you leave this episode today with a renewed sense of possibilities that lie ahead in the intersection of AI and medicine. We hear a lot of doom and gloom stories. I'm hoping that we restored the balance in the universe a little today. And on that positive note, I wish you well. Thank you for listening as always. And hopefully you'll join me again tomorrow. You're all cordially invited. So hopefully I'll speak with you all then. Bye for now.